Hi guys, thank you for coming to our topic 8 review session. This is going to be regulatory pathways in SG2 and M phase, and we're going to start with an overview of the cell cycle. So we start with G1, which is gap phase, where the cell prepares for DNA replication, make sure they have enough nutrients, cell machinery, no mutations. This is where they hit the restriction point where they commit to the cell cycle. Then we have S phase, which is DNA synthesis, G2, which is the second growth phase where the cell preps for chromosome segregation, and M phase. There's also a checkpoint during this phase. M phase is chromosome segregation. So we have prophase, metaphase, anaphase, and telophase. We also have a very important metaphase checkpoint here. Then a quick review of CDKs, which are important throughout the cell cycle.
So CDKs are often referred to as the master cell cycle regulators just because they perform a wide variety of jobs. They're serine threonine kinases, and they activate protein complexes required for events in the cell cycle. They're regulated by cyclin levels, phosphorylation and dephosphorylation. And it's also important to note that just because something is being phosphorylated, it doesn't necessarily mean it's being activated. It could also cause it to deactivate. And then you need both activating phosphorylation and cyclin for CDK to be completely active. One isn't enough. And then anytime you reach a checkpoint, the cell brakes are put on by inhibiting or deactivating CDK.
Quick review of cyclins. Cyclins are required for CDK kinase activity. They activate CDK kinase's activity and they tell CDK what to phosphorylate, specificity. And cyclin is only regulated through transcription translation and ubiquitin-mediated proteolysis.
And then next we're going to go into S phase. So when you enter S phase, it's when the pre-IC is created. So after the pre-RC is assembled, other proteins are added to create this pre-IC. This complex does require active CDK for formation. And then after this pre-IC is formed, the polymerase can be recruited. And then once the polymerase is recruited, then that whole complex together is known as the replosome. And then this origin of replication fires and DNA replication can begin. Except not all origins will fire at the same time.
So further into S phase CDKs inhibit new PRCs from forming. You only want to replicate the DNA once per cycle. If you do it more than once, it's bad for the cell. So S-cyclin CDK complexes phosphorylate ORC to prevent binding of CDC6. They phosphorylate CDC6, which leads to the degradation of CDC6, which occurs through ubiquitin-mediated proteolysis. And this makes sure that MCM cannot be recruited. And then cyclin CDK complexes also phosphorylate MCM27 for nuclear export. So all of these processes are just to make sure that pre-RCs aren't reforming and DNA isn't being replicated twice.
Also, just a quick note to relate this back to past topics, just in case there's an exam-like question like this. So since it gets exported out the nucleus, it would be important to note that this means that this has to have a nuclear exit signal sequence. So just trying to relate things back, because in the final everything will be cumulative, so the topics will merge. Okay, so then continuing with S phase, you have the cohesin rings, which are formed around the replicating DNA. They keep these strands together throughout and after duplication into M phase. They prevent the chromosomes from segregating during spindle assembly and ensure an equal number of chromosomes. And these rings need to be present in S phase in order to function properly in M phase for the equal sensing of tension that we'll get to later. And then at the end of S phase, she doesn't go over it that detailed in class. We assume that there is some sort of checkpoint, but we don't know exactly if it's checking for DNA damage or maybe if it's completely replicated or not. And then also it's important to note that P53 activation can happen in this phase as well.
Okay, so going into the second growth phase. During G2, the cell is growing again. It's prepping for chromosome segregation during M phase. It's making sure that the DNA is done being replicated and then it doesn't have any damage. The cell will not enter M phase until the cells are grown and the DNA is checked. And this growth phase is very important because it's right before the cell divides. So very important that everything looks okay. And if there's no activation of M phase CDK, there will also be no entry into the N phase.
Okay, so at the beginning of G2 phase, MCDK levels are rising but are inactive. And then CAK phosphorylates MCDK with an activating kinase, but it's still weaker than the inhibiting phosphate. And then while this is occurring, it's also held inactive by WEE1, which is an inhibitory kinase, which places the inhibiting phosphate on MCDK. And then the reason it has this inhibitory phosphate placed on it is so it could still grow big enough, or else it will just go through prematurely without enough nutrients.
During late G2 phase, after the checkpoint, where it makes sure everything looks okay, the cell will activate MCDKs to proceed to M phase, so producing those M cyclins. CDC25, an activating phosphatase, is partially activated by PLK1 kinase. So that takes off the inhibiting phosphate on MCDK that WEE1 had placed on. So now that that inhibiting phosphate has been taken off, MCDK is active. So then the active MCDK goes through a positive feedback loop. So that can continue to produce more of the CDC25 phosphatase to continue to take those inhibitory phosphates off. Yeah, so it's just kind of a enhanced event.
Does anyone have any questions before we get into M phase?
I think by that it means that's like the beginning. So when it's partially activated, it goes in and starts to take off inhibiting phosphates. And then once MCDK is activated, it goes in and upregulates the production of CDC25. So then it can continue to take off those phosphates at a higher level. Through the positive feedback loop.
Great. Thanks. And then this is just a little quick overview of M phase, but we'll go into detail more as we continue with the slideshow. But first, it's prophase. That's where the nuclear envelope breaks down. You have pro-metaphase. That one we don't focus too closely on in this class. Then metaphase, where they align in the middle. That's where the spindle assembly checkpoint is. Anaphase, they get segregated. And then telophase and cytokinesis, where the envelopes start to reform and the cell divides.
So during prophase, the first step of M phase. So we have active MCDKs, which phosphorylate lamins to break down the nuclear lamina. CDKs also phosphorylate condensin to induce chromosome condensation, where the spaghetti turns into a sausage form. So those chromosomes are condensing. They phosphorylate various MAPs and motors to facilitate mitotic spindle assembly. So just getting everything ready.
And then we also just have sequential phosphorylation as a quick side note. So the main idea is that phosphorylation needs to happen in a certain order. And an example of this relating back to, I think, topic 5 is the sequential phosphorylation of AKT in the survival signaling pathway. And then just another quick reminder, CDKs are the master cell cycle regulator, and that CDK can act as a priming kinase for downstream kinases, and that inhibiting CDK will prevent cascade and halt cell cycle in any phase needed. So then this is like back to PLK1. So substrates of this need to be phosphorylated by CDK in order for PLK1 to bind to the substrate.
Okay, so going into spindle assembly, microtubules reassemble into spindles and separate chromosomes during the beginning of M phase. So types of microtubules, we have asters, which grow from spindle poles and attach to cell cortex. Kinetochores grow from spindle pores and attach to kinetochores of sister chromatids. And then interpolar microtubules grow from spindle pores and overlap at the middle.
And then just a quick in-depth or overview, I guess, of the kinetochore. They're just large multi-protein complexes that are built on the centromere of each sister chromatid, and it's composed of about 80 different proteins. And the spindle microtubules attach to these specific proteins within the kinetochores.
So opposing forces from motor proteins, push and pull forces, are at work during spindle formation. So spindle formation requires an array of opposing forces, where kinesins are plus force and dyneins are negative. So they walk towards opposite ends of the microtubule to create opposing pulling forces that stabilize the spindle. So EG5 is an example of a motor protein that binds interpolar microtubules that overlap. They're plus end directed, and they push spindle poles farther apart.
And then here's just a little diagram of how everything works. And the dyneins, which are minus end directed, they hold the cell cortex, which is the inner layer of the membrane, and walk towards the minus end of the microtubule that creates the like pull mechanism. And then a combination of these different motors in different places throughout the spindle network is what creates the structure and proper preparation to pull these chromosomes apart.
So going further into the push and pull mechanism, so interpolar microtubule forces, there'll be EG5, which is double-sided. They're homotetramers. They'll be walking to the plus end because they're plus end directed motor proteins. They push spindle poles apart and they walk toward the plus end on two overlapping microtubules simultaneously, which causes that pushing apart. And then dynein is minus end directed, so it pulls the spindle poles together and walks toward the minus end of one overlapping microtubule while holding on to the other overlapping microtubule. And then aster microtubules involve dynein, which is minus end, spindle pulls apart, where one end is attached to the cell cortex, which is the area just inside the cell membrane, and the other walking end is along aster microtubule towards the minus end.
And then the next we'll be talking about is metaphase. So here, the sister chromatids are still held together by the cohesing rings that were added in S phase, and they're also attached to the microtubules at the kinetochore. And then you have equal and opposing pulling forces that cause the lineup of chromosomes along the metaphase plate.
So we have metaphase to anaphase, which is the spindle assembly checkpoint. So this checkpoint ensures that all sister chromatids are aligned and that spindles are attached properly at the kinetochore. So the cell will inhibit anaphase until there's proper microtubule attachment on the kinetochore, and otherwise non-disjunction or uneven separation of chromosomes will occur. So the main ideas about MAD, BUB, and CDC20. MAD and BUB sense microtubule tension on the kinetochore, and if there's equal tension, CDC20 will be released, which allows the cell cycle to move forward.
And then this is just the mechanism of MAD bub CDC20. So CDC20 is released and it binds and activates APC, which is a ubiquitin ligase. Then this complex will target the protein securin for ubiquitination. Securin is currently bound to separase, which inhibits separase's function. So then once securin gets degraded, separase is free and it's active. And then it can then cleave cohesin rings. And so then the sister chromatids can separate, which is how it enters anaphase. And then also a side note, APC also targets M cyclin for degradation. So the M cyclin CDK will begin to drop in activity.
So now we are in anaphase. So anaphase A, chromosomes are pulled poleward or to opposite ends of the cell. Chromosome is walked by minus end directed motors. The kinetochore microtubules shorten and the spindle poles are still in place. And then into anaphase B, the spindles will lengthen. There will be microtubule growth on the plus end and the poles will be pushed/pulled apart. There's a sliding force between interpolar microtubules from opposite poles, which is a pulling force directly on poles to pull them apart.
So then lastly, we have telophase and cytokinesis. So at this point, the sister chromatids are on opposite poles. And then you have cleavage furrow, which is created by a contractile ring. And then indentation in cells membranes that gradually deepens until it hits the spindle that is left between the two nuclei, and then they eventually split. And then also, again, just remember that M cyclin CDK activity has dropped by this point. The spindle disassembles, and then the chromosomes decondense to form chromatin. So it's going back from like the sausage-like structure to spaghetti, and then the nuclear envelope reforms.
So just to summarize with some main ideas, the cell cycle events happen in a strict sequential order, G1, S, G2, M, which are further broken up. Think of M phase. Cell cycle events include cyclical activation and deactivation of proteins, often by means of phosphorylation with fresh ATP. CDK must be bound to cyclin and phosphorylated in order to be active and not be phosphorylated by WEE1. And CDK regulates various proteins in different phases of the cell cycle. And different phases of the cell cycle have their own cyclins, which are created when needed and degraded depending on the part of the cell cycle where they're needed. And regulation for proper functioning requires activation and deactivation and inhibition. So don't think of them as more positive or negative than the other.
Does anyone have any questions about M phase? No? Okay. Well, that's all we have for today.
Let us know if you have any problem set questions or anything like that.